Hello, everyone, and welcome. Today, we're diving deep into the most basic, most fundamental building block of every single living thing on our planet. We're talking about everything from the tiniest bacteria to the largest blue whale. And yes, that includes you and me. We are all made of cells. So let's get started on our journey into this incredible microscopic world.
So here's what we've got on the agenda today. We're going to start with a little bit of a history lesson, traveling back to when cells were first discovered. Then we'll map out their basic structure. After that, we'll take a tour inside a bustling cellular city to see how everything works and find out how cells eat and breathe. We'll even walk you through a hands-on experiment you can do. And finally, we'll wrap it all up with a quick review to make sure you've got it all down.
All right, let's begin. Our story starts more than 350 years ago, in a time when so much about the natural world was a total mystery. No one really knew what living things were made of. And, the breakthrough didn't come from some grand, complex theory. It all started with a simple look through a new invention.
This really is the big question, isn't it? If you could just zoom in and in and in on a leaf or your own hand, what would you eventually find? What is that basic little unit that makes something, well, alive? The answer to this question changed biology forever.
And here we have our first clue. This comes from a scientist named Robert Hooke way back in 1665. He was looking at a super thin slice of cork through his homemade microscope. And what he saw amazed him. It wasn't solid at all. Instead, it was made of tons of tiny box-like compartments. They reminded him of a honeycomb, or the little rooms, the cells, where monks lived. So he called them cells, and the name stuck.
What's so cool about this timeline is you can literally see our understanding grow, piece by piece. So after Hooke saw those empty cork cells, another scientist, Leeuwenhoek, built a better microscope and saw the first living cells, tiny little creatures swimming around in pond water. Amazing. Then, much later, Robert Brown spotted the nucleus inside a cell. All this groundwork led two scientists, Schleiden and Schwann, to propose the cell theory, basically that all plants and animals are made of cells. And the final, crucial piece of the puzzle came from Virchow, who declared that every single cell has to come from another cell that's already there. Life, it turns out, comes from life.
So, we've got the history down, but what is a cell, really? Let's move on and look at its basic design. Turns out, whether you're looking at a cell in an onion or a cell in your own body, most of them share three fundamental features.
Okay, I want you to think of a cell like a tiny, self-contained factory. It's got an outer wall with security gates, which we call the plasma membrane. This wall controls everything that comes in and goes out. Then inside, you've got the main office, or the control center. That's the nucleus, which gives all the orders. And the entire factory floor is filled with a jelly-like material called the cytoplasm. These three parts are the foundation for pretty much every cell. Now, that cytoplasm, it's not just some empty goo. Oh no. It is a super busy, bustling place. So let's imagine the cell is a miniature city. This city is packed with tiny structures called organelles, and each and every one has a specific, super important job to do. And as you can see right here, not all of these cellular cities look the same. On the left, you've got an animal cell. On the right, a plant cell. Just take a look. Can you spot some differences right away? The plant cell looks a lot more rigid, more rectangular, right? Like a little brick. And do you see those green things inside? Let's take a closer look at what makes them different. Okay, here's the breakdown. Plant cells have three main things that animal cells just don't. First, on the outside of their membrane, they have a tough, rigid cell wall. It's like a building's frame, giving it that fixed shape. Second, they usually have this huge central vacuole. Think of it like a giant water storage tank that keeps the cell firm. And third, they have chloroplasts. These are the tiny green solar panels that make food for the plant.
Let's go back to our city analogy because it really helps make sense of all these parts. The mitochondria, they're the power plants generating all the energy the cell needs to function. The endoplasmic reticulum is the city's highway system, transporting materials all over the place. You've got lysosomes, which are like the waste disposal and recycling crew, breaking down old junk. Vacuoles act as storage warehouses. And running the whole show from City Hall? That's the nucleus, directing every single activity.
Okay, so we've got a city full of workers, but how does the city get its supplies in, and how does it get its garbage out? Well, everything has to go through that plasma membrane, the city's border. Let's see how that works.
The main way things move is through a process called diffusion.
It's really simple. It's just the natural tendency of things to move from a crowded place to a less crowded place, to spread out. You know, if you light an agarbatti in one corner of a room, the smell doesn't just stay there, right? It spreads out until you can smell it everywhere. That's diffusion. For a cell, it means that when waste like carbon dioxide builds up inside, it naturally moves out of the cell to where there's less of it. Easy.
Now, you might hear the word osmosis. Don't let it scare you. Osmosis is just a special name for the diffusion of water. That's it. When water moves across the cell membrane, which is a bit picky about what it lets through, we just call it osmosis. This is a super important process. It's exactly how plant roots absorb water from the soil. The water's just trying to balance itself out on both sides of the membrane.
So this affects the cell in different ways. Look at this. In what we call a hypotonic solution, like plain water, there's more water outside the cell, so water rushes in, making the cells swell up. Think about putting dried raisins in water. They get all plump. In a hypertonic solution, like really salty water, the opposite happens. There's more water inside the cell, so it rushes out, and the cell shrinks. And finally, in an isotonic solution, it's perfectly balanced. Water moves in and out at the same rate, so the cell stays the same size.
Okay, now for the really fun part. All this theory is great, but there's nothing like seeing it for yourself. So let's walk through the steps of a classic experiment that you'll definitely do in your science class, preparing a slide of an onion peel to see real living cells. And to do this, we're going to need this amazing piece of equipment, the compound microscope. This incredible tool is our window, our portal, into a world that's completely invisible to our naked eye. All right, ready? Here's how you do it. First, you take a piece of onion and carefully peel off that super thin, transparent layer from the inside surface. You want to pop that right into a drop of water so it doesn't dry up and curl. Next, you gently move it onto a clean glass slide that has a drop of water on it. Now, to make the parts of the cell actually visible, you add a drop of a stain, like safranin. And for the final and trickiest part, you gently lower a thin piece of glass called a cover slip over it. Try to do it at an angle to avoid trapping any air bubbles. And there you have it. If you did it right, this is what you'll see under the microscope. A beautiful, orderly pattern of rectangular cells, all lined up like bricks in a wall. You can clearly see the strong cell wall, and that dark little dot inside each cell, that's the nucleus we've been talking about, the city hall. Wow, we have covered a lot of ground today. So before we wrap up, let's do a quick review. We'll go over some key questions from your textbook to make sure all these new ideas are locked in place. Okay, first question, and it's a popular one. I'm sure you remember our city analogy. Which part of the city was the power plant, the one that generated all the energy to keep things running? And more importantly, why do we call it that? And the answer is the mitochondria. They're called the powerhouses because they take in nutrients and oxygen and, through a chemical process, release energy. This energy is stored in a special molecule called ATP. Now, the best way to think of ATP is as the cell's money, its energy currency. To get any job done, whether it's building something or moving something, the cell has to spend some ATP. Without the mitochondria making this currency, the entire cellular city would shut down. Let's tackle another really important idea. We need to understand the two major types of cells that exist in the world. So, what are the biggest differences between a simple prokaryotic cell, like a bacterium, and a more complex eukaryotic cell, like the ones that make up plants and animals? The single biggest difference really comes down to organization. Eukaryotic cells, like ours, have a true nucleus. That means their genetic material, their DNA, is neatly contained and protected inside a membrane. Prokaryotic cells are much simpler. Their DNA just kind of floats around in a region called the nucleoid. On top of that, eukaryotic cells have all those specialized membrane-covered organelles we talked about, the power plants, the highways, the whole city infrastructure. Prokaryotes, they are much more basic and are missing most of those complex parts. And we'll end with this thought. We've come a very, very long way from Robert Hooke's little rooms to our incredibly detailed map of the cellular city. But you know what? The journey of discovery is never, ever over. If a single cell is like a whole universe in miniature, just imagine the secrets that are still waiting to be discovered inside. What do you think the next big breakthrough will be? That's something for you to think about. Thanks for joining me today.